Hello, my name is Jeff Dunne from the Department of Crop and Soil Sciences at North Carolina State University, working in peanut breeding and genetics. Today I would like to present some of our initial work on characterizing leaf spot disease through image analysis of advanced breeding and germplasm lines within our peanut breeding program. The Peanut Research Foundation, which is the research arm of the American Peanut Council, estimates that the annual losses due to early leaf spot caused by Passiflora racqueticola and late leaf spot caused by Northopassiflora personata is around $53 million per year. To control leaf spot in the Virginia Carolinas region alone, it is estimated that peanut farmers spend around $80 to $90 per acre, which equates to $17 million per year when considering the average production acreage. Some of the primary concerns regarding leaf spot and the resistance to these diseases is the pathogen overcoming plant resistance, some chemical resistance, and shifts from one pathogen to another. There seems to be a limitation on novel chemistries and more of an emphasis on combination fungicides, which has the potential of allowing the pathogens to develop resistance to multiple fungicide modes of action. From a plant breeding sense, we cannot control this level of resistance, but from a host level resistance, we can identify novel resistance genes within our germplasm and breeding lines that can help provide some protection if fungicides start to fail. In addition, if a resistance gene is only effective against one or the other disease, shifts in pathogen populations year to year can easily overcome resistance genes specific to one or the other. Peanut Research Foundation, along with the National Peanut Board, joined together to provide funding for the Peanut Genome Initiative. The goal of this initiative in Phase 1 was to provide a reference genome sequence of the progenitor species and cultivated species to benefit applied peanut breeders in the development, selection, and release of new cultivars. This phase was completed in 2019 with the release of the Arachis durinensis, Ipaensis, and Hypogaea genomes. In Phase 2 of the Peanut Genome Initiative, the Peanut Research Foundation is funding 4 pillars of applied research in an effort to utilize the reference genomes produced in Phase 1 to develop germplasm and cultivar releases for peanut improvement. The areas of interest under the Phase 2 directive is improvement of disease resistance, aflatoxin research both pre- and post-harvest and storage, drought tolerance, and flavor improvement. For this research, we fall under disease resistance as we're trying to improve and release cultivars with higher levels of leaf spot resistance, which was their main emphasis for this category. Within our breeding program, we have varying levels of resistance depending on the resources and the specific program. In the cultivated breeding program, the one that results in cultivar releases to growers, we have a wide range of resistant and susceptible breeding lines. On the left, this is a comparison of the older cultivar NCVA 92R and a newly developed breeding line N16021. Relative to the performance in the cultivated breeding program, the wild species breeding program has way more resistant germplasm lines. On the right, the line in the center is a newly developed interspecific hybrid between an older cultivar Gregory and a wild species diploid Arachis diagoi. The level of resistance is greater than any line in the cultivated breeding program, however the agronomic properties are extremely low. On an overall view of the two breeding programs, there is clearly a difference in the level of resistance to leaf spot represented in these drone images. On the left is our advanced leaf spot trial. Clearly a number of lines are brown and completely defoliated under intense leaf spot pressure with no applications of fungicides. On the right, the wild species shows consistently high levels of resistance to leaf spot under the same leaf spot pressure with no applications of fungicides. These two images were taken on the same day. The main sources of resistance that the cultivated breeding program is using for early and late leaf spot resistance comes from Arachis cardnacii and Arachis diagoi. In the development of the cultivar Bailey, a wild species introgression line from Arachis cardnacii was used to develop N03081T, which eventually became Bailey. So we know that it is possible to transfer these beneficial resistance genes into superior yielding lines in the cultivated breeding program. Currently, our efforts to identify and select for leaf spot resistance are somewhat limited to a subjective evaluation of breeding plots by the peanut breeder. These ratings aren't as straightforward as you'd think, and often can be compromised by changes in weather, raters, replications, field size, etc. For leaf spot, we rate on a 1 to 9 scale, where 1 means the line is resistant or doesn't have any visual lesions or defoliation. 5 means it has significant leaf spot lesions, but no noticeable defoliation. And then 9, which means the plot has been completely defoliated or reduced to vines. In the image to the right, I have a peanut leaf that shows symptoms of early and late leaf spot. Early leaf spot is depicted as having light brown lesions with a yellow ring or a halo. However, the halo isn't always present. Late leaf spot are more darkly colored and generally lack a halo with prominent sporulation on the underside of the leaf surface. When taking ratings on the plot level, it's incredibly difficult separating out early versus late leaf spot percentages visually. Therefore, our ratings simply become an overall percentage of lesions. When it comes to taking ratings on plots, typically the ratings are relative to other plots. When I take a rating on leaf spot, I usually walk the entire field and get a mental representation of what I would consider to be a 5 rating. I then continue looking at the plots until I'm confident I could identify all ranges of the rating scale. I'll give you an example. Here I'm suggesting the plot on the far right is considered a 5. With only knowing this rating, could the ratings of the other plots be identified? Here are my thoughts. Based on the rating of a 5, I might rate this plot a 3 in comparison. The plot is healthy and had minor symptoms of leaf spot. The one next to it is nearly completely defoliated, so I'll rate it an 8. The final 2 plots I would rate a 4 because of a slightly higher leaf spot lesion percentage, and the one in the middle, the Arachis diagoy line, I would probably rate a 1 because it has no visual symptoms of leaf spot and would be considered resistant. The rating may differ due to changes in weather, between raters, or across the field, because we're human. In order to get a consistent representation of leaf spot infection, a high-throughput method of evaluation needs to be developed. The overall importance of taking good, consistent ratings is that it helps us to map the genes responsible for resistance so that we can design genetic markers that will identify families and/or lines with those important genes, and then use them in our program to make selections. On the left is a representation of a single introgression block and multiplexed CASP or PACE assay that we use for selection purposes within our program. For a single assay, we can select for fixation in green or enrichment, yellow, for the next generation of development. For an introgression block, we design flanking markers with similar fluorescence patterns after the same number of PCR cycles to bring introgression blocks conferring resistance from the wild species program into the cultivated program. And finally, we can stack multiple single marker assays together with similar fluorescence patterns after the same number of PCR cycles to select for multiple traits. On the right is a simple schematic of the NCSU Peanut Breeding and Genetics Program's breeding pipeline. We make crosses and we send those to Puerto Rico to get an extra generation of development. When the lines come back, we progeny test them in the field for disease resistance, in this case leaf spot resistance. We identify the best families from the field and pair it with the marker results, select the plants with the best pods for those families, and then repeat the cycle. After several cycles, these lines then advance to our yield trials and hopefully one will be selected for release. So based on this information, we developed a project with the following objectives. The first is to identify objective methods for evaluating leaf spot, both early and late leaf spot, for improving phenotyping and data quality. The second is to produce a high throughput protocol for objective methods for evaluating leaf spot. The third is to evaluate and genetically map early and late leaf spot resistance among the breeding families and lines in the North Carolina State University Peanut Breeding and Genetics Program. And finally, use these identified genetic markers to select families and lines in the cultivar development program with increased resistance to leaf spot, and this can be through marker-assisted backcrossing or selection. For the purposes of this presentation, we'll be reporting results for the first objective only, as this is an ongoing project. The data that I'm going to show today represents some of the preliminary work that was conducted in 2019 and 2020. In 2019, representative samples of commonly grown cultivars within the breeding program leaf spot trial were assessed using developed scripts in FIJI, which is just ImageJ, to separate the visual rating into their subcomponents. So what we wanted to know is, one, could we effectively scan the tissue collected and differentiate between early and late leaf spot using the images alone? And in addition, could we then use the tissue scans to estimate an overall percentage of leaf spot infections on leaf tissue? And then two, could we track using UAV imaging over time the relative canopy loss due to leaf spot infection that could be attributable to defoliation? To do this work, 8 leaves were randomly selected per plot, and then the 32 leaflets were scanned on an Epson scanner for analysis. The canopy cover was captured using a DJI Mavic 2 Pro drone, which comes stocked with a 1-inch sensor, and the images were stitched together using Agisoft's Metashape. So it turns out it's way more difficult to separate these two diseases using basic thresholding. The issues include, one, whether each leaf spot can be accurately identified and how long it takes to go through each leaf sample. Two, the resolution in the image may not allow us to accurately differentiate the spots. Three, the thresholding cutoffs that sort the clusters, leaf spots, may be too similar. And four, the population pressure of each disease in a given year may change. For example, the peanut pathologist here estimated that late leaf spot infection was around 95% in 2019 among the two diseases. In order to attempt at differentiating these diseases, more data collected on the specific leaf spots is needed in order to develop a better training model for accurately classifying early and late leaf spot. That being said, getting a percentage of lesion area is pretty straightforward. The FIJI script is designed to threshold the leaf spots and the entirety of the leaf surface separately. The ratio between these two pixel totals gives us our leaf spot estimates on the leaf surface. The script is automated to run through each of the images collected per plot and can calculate the final leaf spot percentage. Similarly, the drone images help us to estimate canopy cover changes during heavy leaf spot pressure. The top and the bottom rows of images represent pre and post leaf spot onset respectively. In the top row, images were taken on September 6th, and in the bottom row, images were taken on October 8th prior to harvest. The first column shows the thresholding across all of the plots. The second column shows a similar image crop to our gridding of each individual plot, and the final column shows the final image threshold. Using these two canopy scores, we can estimate a change in canopy cover that we're calling defoliation. In some instances, when a plot is relatively unaffected by leaf spot, the defoliation score can actually be negative due to the continual or unaffected growth. We can then apply this method to our entire breeding program. In the field, we grew out 220 lines in 2020 and used this approach to get leaf tissue and defoliation estimates for each plot on a bi-weekly basis during heavy leaf spot pressure. We additionally have yield data collected on these untreated plots, along with plots that were treated with fungicide, but this data will not be presented here. Presented are the results obtained using the previously described methods collected in 2020 on the 220 breeding and germplasm lines selected from the program. On the left is a figure of 26 lines that were whole genome sequenced for another project for developing a high-throughput genotyping platform for our program moving forward. These lines were chosen based on several traits of interest, including resistance to leaf spot. These differences in the bar chart represents the visual ratings taken on these genotypes prior to harvest. On the right are some basic statistics describing the different evaluation methods, including the tissue scans alone, the drone or defoliation score alone, or a combination of the two using the first principal component when compared to the visual scores. In each instance, the evaluation type was highly correlated with the visual rating. In addition, the visual score, which again is a 1 to 9 rating, was also modeled using logistic regression to determine the predictive potential of each evaluation type. The logistic model produces overall concordance scores, which is the model's predictive potential, and odds ratios for each evaluation parameter. The concordance scores compares pairwise scores across the dataset to determine if the probabilities associated with being in a higher visual score are true for each paired test. For the odds ratio, this is determining the odds of being in a higher visual score given an increase in the evaluation unit. If you look at the models evaluated, tissue alone, drone alone, PC1, and the full model of tissue plus drone, there is an increase in concordance when considering more parameters. But it seems the PC1 that explains around 79.1% of the variation is similar to the full model. That being said, there isn't much of a difference in concordance when using tissue or defoliation data alone either. The odds increase with the number of parameters too. For tissue alone, there is 1.3 times the odds of being in a higher visual score when the percentage of leaf spot surface area increases, which makes sense. Under the PC1, we're 10 times more likely to be in a higher visual score when the two components, tissue and drone, increase together. Our future work on this leaf spot evaluation method is funded through the USDA NIFA plant breeding program, and the work over the next 2 field seasons will be conducted by my PhD student, Cassie Newman. The work that we're doing in the field this year consists of multiple rating systems, timings, and data collection types. I will be evaluating the plots for leaf spot on the previous 1 to 9 scale. This will be done in a weekly manner. Cassie will be evaluating the plots using the low-throughput objective method of drone imaging for defoliation and tissue collection for lesion counts and leaf spot differentiation. Lastly, we'll also be collecting the high-throughput images and developing an algorithm to rapidly analyze the images for instant leaf spot ratings using cell phone capture. The collection timing will essentially be from August through harvest in October. Here we're diagramming our high-throughput method that just uses drone and/or cell phone images to rapidly take ratings on both defoliation and leaf spot lesions. The idea here is to evaluate the 220 lines in the field using drone imaging and concurrently walk through the plots taking cell phone images. The images will be instantly uploaded where an image software with a customized algorithm will be waiting to analyze the plots. In addition, we're going to evaluate using Canopio to determine the effectiveness of this application in detecting leaf spot lesion counts and canopy changes. Using these approaches, ratings can be objectively taken and in less time. If the analysis is completed, I can then look at the data and make selections within these lines for further development. Ultimately, what we're hoping to achieve is to pair our newly developed genotyping pipeline, which capitalizes on whole genome sequencing and in silico digestion for optimized GBS, developed by Cassie Newman, to high-quality leaf spot ratings in order to identify regions or introgression blocks that are influencing leaf spot resistance. With that, I would like to thank all of those organizations that sponsor our research, particularly the National Peanut Board, the Peanut Research Foundation, Severn Peanut Company and Hampton Farms, and the USDA ARS and NIFA programs, and I want to thank you for your time. If you have any questions regarding this project, please feel free to ask, or you may call or email me. Thank you.